Hello, I'm Jiwon Lee from South Korea, and I'm in grade 11 in Chadway International. Today, I would like to introduce the novel gene therapy targeting ZGPAT that leads to a faster wound healing in human skin cells. I have developed my interest in curing the human body. Last year, I filmed and edited an exercise video that can reduce spinal pain for scoliosis patients and published it on YouTube to make it accessible to the public. A few months ago, I created a website on fasting that contains information on the benefits and types of fasting and the process called autophagy that takes place during fasting. Similarly, I wanted to extend my knowledge on curing the human body through the wound healing investigation. First of all, I hypothesized that when EGFR is activated by targeting the ZGPAT gene through RNA interference, or RNAi, the rate of cell migration would increase. To test this hypothesis, I targeted the ZGPAT gene, which acts as an EGFR repressor, in the Detroit 551 human skin cell and measured the rate of cell migration through the wound healing assay. The research was conducted in mainly 7 steps, starting from Detroit 551 cell culture, siRNA transfection, total RNA extraction, cDNA synthesis, polymerase chain reaction, agarose gel electrophoresis, and scratch wound healing assay. Through this experiment, I was able to find the optimal annealing temperature for each primer through multiple PCR reactions. 50 degrees Celsius was the annealing temperature for ZGPAT and 57.5 for EGFR. As a result, when the cells treated with siRNA ZGPAT were compared to cells treated with siRNA negative control, about 20% reduction in ZGPAT gene expression and about 500% amplification in EGFR gene expression were observed. Next, the wound healing assay was conducted to observe whether targeting the ZGPAT increases the rate of cell migration. 4 samples, as shown in the photos, were prepared. The result concluded that inhibiting ZGPAT with EGF treatment activates the cell migration. I used a program called ImageJ to quantify the rate of cell migration by quantifying the cell-free area. As shown in the photos, the yellow line is the boundary line that distinguishes the cell-free area. The rate of migration was found by dividing the area without cells at 0-hour mark by the area without cells at 24-hour mark. The results showed that when ZGPAT was suppressed in the presence of EGF, the migration rate increased to 2-fold. In conclusion, when siRNA targeting ZGPAT and EGF were treated together, the rate of cell migration increased. This conclusion shows that the sensitivity of EGF, a protein that is commonly found in wound ointments, can be increased by siRNA targeting ZGPAT. This experiment presents a more efficient method for wound healing by inhibiting the ZGPAT to activate the EGF-EGFR signaling in order to increase the cell migration. Thank you.